The COVID-19 vaccine was created with brand new technology that we know nothing about. Hashtag not
Getting the
safe.
COVID-19 vaccine gives you COVID-19. I was
I got the
so sick
COVID-19
after getting
vaccine,
both
but I
my
somehow
injections.
still made my little
Hashtag
The
sister
not
COVID-19
worth it.
vaccine was
Obviously, it doesn't
created
stop
way
you from
too
getting
Why would I
COVID-19.
need to
There's
get
no
the
way
vaccine
that
if
if
I've
you're
already
supposed to
had
get one
COVID-19?
Pfizer dose and one
Hashtag
Moderna
you're
dose because
pointless.
they'll be mass
boosters
protected.
forever.
The
Hashtag
COVID-19
again.
vaccine is going to
Hashtag
change
I
my
don't
DNA.
understand why there are
Hashtag
multiple
FYI,
mRNA
you're going to test
vaccines.
positive
Aren't
on
I
Pfizer
want to have
and
PCR
children in a
Moderna
few
test if you
years.
get a
So
COVID.
there's
Whoa, hold on. I know there's a lot of information out there, and it's hard to determine which are reputable sources. It can be quite overwhelming, but lucky for you, as medical genomics master's students at the University of Toronto, my classmates and I prepared an informative webinar about mRNA vaccines. With access to the proper resources to correctly inform you, we'll be going over what mRNA is and how it can be turned into a vaccine, the history of mRNA vaccines, coronavirus and the COVID-19 vaccine, differences between the two headlining mRNA vaccine companies, Pfizer and Moderna, and debunking the current myths that are circulating. Hopefully by the end of this talk, you'll be able to understand the complexities surrounding this pandemic and form your own judgments about distinguishing myths from truths. So come along and let us walk you through it. Let's get right into it, starting at the simplest level. Human cells contain DNA, RNA, and proteins. DNA stands for deoxyribonucleic acid. And let's break that up really quickly. Deoxyribose means there is a 5-carbon sugar molecule that has lost a specific oxygen group. And nucleic acid refers to the molecule made of 5 different types of nucleotides, which are just combined compounds containing the deoxyribose sugar group, a triphosphate group, and a nitrogenous base. These nitrogenous bases ultimately determine if the nucleotide is an adenosine, cytosine, guanine, thymine, or uracil in the case of RNA. It's a unique combination of ACTGs that generate what you may have heard it being called the code of life. However, DNA cannot act as the code of life on its own. DNA only contains the code to create all the proteins your body needs to function properly. And it creates these proteins using a special form of RNA called mRNA. RNA is similar to DNA and stands for ribonucleic acid. The M in mRNA stands for messenger because mRNA communicates the DNA code to the machinery that builds proteins. In a process we call transcription, an enzyme called RNA polymerase II reads DNA and uses nucleotides, specifically A, C, G, and U this time, to build the mRNA. In the next step, which we call translation, specialized RNA molecules called tRNA read the mRNA and string together the building blocks of proteins, called amino acids. A string of amino acids is called a polypeptide, which is the same thing as a protein. No, this is not the same as the protein you may be thinking of. And if this still sounds a bit confusing, let us describe this using a different analogy. If you want to build a new house to live in, you will need the blueprint, which contains all the special instructions, as well as the architects for reading the blueprint and interpreting what building material is needed. They will then pass this information onto the construction workers so they can build a structurally sound house. Feel free to pause the video here to match the DNA, RNA, and enzymes to their analogy counterparts. In this example, DNA is the blueprint, the architects are the mRNA, the building material is the amino acids, and RNA polymerase II and tRNA are the construction workers. The cool thing is that when our cells are done with mRNA, they recycle the materials to make new mRNA later.
Wow, how efficient!
To do this, a team of enzymes, which are just specialized proteins, chop the mRNA into tiny, non-functioning pieces, which frees the nucleotides in the cell for the next transcription process. Now that we have covered the general foundation of genetics, it's easy to see how the creation of proteins from mRNA gave scientists the idea to use it as a drug, because it can enter our cells and create proteins to help us get better. One strong application of this stems from the fact that mRNA can create proteins that mimic foreign substances, which allows for the creation of antibodies. Naturally, our bodies process these substances through our immune systems. The immune system not only confronts them directly, but also creates antibodies specific to the type of substance it encountered, so that if it ever infects you again, your immune system will have the memory to react quickly and protect you from it. We call this acquired immunity.
Acquired immunity.
Now, by utilizing this mechanism, researchers developed a new type of mRNA-based vaccine, which has been rapidly applied in the past decade. The core of the mRNA vaccine is a modified mRNA, which is coated by lipid nanoparticles in order to be delivered into the human body. Lipid is just a special word for fat, and these lipid nanoparticles form a fatty envelope that surrounds the mRNA. These fats are the same kind that also surround our cells, so the vaccine and our cells easily fuse together and the mRNA is left inside the cell. Once inside, it goes to the endoplasmic reticulum, a specialized cellular organelle which allows this piece of mRNA to be read and interpreted, just as the other DNA in the cell would be. In the case of a virus, the mRNA vaccine would produce a protein called an antigen, which is harmless and can stably float inside the body. Once these antigens are recognized by special immune cells, the immune system generates antibodies complementary to the viral antigen, so that the body will be able to identify and attack the virus if infection ever occurs. But don't worry, we will cover this in more detail when we go over the coronavirus. Traditional attenuated viral vaccines use the deactivated version of the virus to promote acquired immunity, and include examples like the influenza, MMR, hepatitis B, and HPV vaccines. mRNA vaccines, on the other hand, utilize the RNA sequence encoding the virus's antigen, also called an epitope. Epitopes are highly variable and specific binding regions for antibodies. For example, coronaviruses are a kind of virus that are covered in spike protein epitopes, which recognize ACE2 receptors on our lungs, allowing them to attach and make us sick. A couple of benefits of mRNA vaccines over attenuated viral vaccines include rapid production, easy adjustment to new sequence variants, low costs, and less safety concerns involved in the chemical deactivation process. With the use of mRNA vaccines, we would not need to get physically sick first to get these antibodies. We would just create our own. A really prevalent example of that right now is the COVID-19 vaccine.
The COVID-19 vaccine was created with brand new technology that we know nothing about. #not safe.
Oh, what? I thought I turned my phone off. Alright, well, let me answer this anyways. This is actually not true. Let's explore the history of mRNA vaccines over the last 30 years and how we've been studying mRNA itself for 60 years. mRNA was first discovered in 1961 by a team of scientists who found that mRNA is made from DNA to create proteins. In 1978, scientists realized that if they surround mRNA with the same fats that surround our cells, mRNA can be transferred into our cells, and they called this fatty envelope a liposome. By 1992, mRNA was being tested as a treatment for diabetes and influenza in mice. Its therapeutic potentials were further realized when they changed the normal uracil in mRNA with pseudouracil, which hid the external mRNA from the human immune system. In 2012, several different groups of scientists researched using mRNA wrapped in a liposome as a vaccine to protect mice from both influenza and rabies. In 2018, patrazirin, a liposome-surrounded RNA drug, was successfully approved and released to the public. In 2019, a novel coronavirus made its way into the world to be the greatest challenger to mRNA vaccines yet.
That's for sure.
This novel coronavirus, labeled SARS-CoV-2, is very efficient at attacking our lungs due to the location of the ACE2 receptor and causes severe acute respiratory syndrome, which is how it got its name. However, the actual disease associated with the acute respiratory illness was called COVID-19 due to the timing of its discovery. I know the terminology can be confusing, and people commonly mix up the two. So to help you understand the difference, we can compare it to another virus. Just as how HIV can cause AIDS, SARS-CoV-2 can cause COVID-19. 1 year later, several companies harnessed liposome mRNA technology to create COVID-19 mRNA vaccines. Once you receive the vaccine, it can protect you against developing COVID-19 by creating antibodies targeting the virus's spike proteins. When it becomes covered in these antibodies, it cannot attach to our lungs and it has now become neutralized, which prevents it from reproducing and gives the immune system time to destroy the virus.
Getting the COVID-19 vaccine gives you COVID-19. I was so sick after both my injections. Hashtag not worth it.
This is also false. Getting the COVID-19 vaccine gives you the mRNA code you need to create antibodies against the coronavirus to prevent you from getting COVID-19. While you may feel a little sick after receiving the vaccine injections, you don't ever actually get infected with the coronavirus. You feel under the weather because your immune system is responding to the foreign material that was injected into your body. We've come up with a simple analogy to try and help explain this phenomenon. Imagine that there is a castle, and this castle has its own defense system, such as an army of guards. Now imagine one day, a good knight comes to the front gates to warn the people of the castle and relay the message that there may potentially be an incoming attack involving a new chemical. The knight also hands them instructions for making new types of weapons that will specifically deal with this kind of attack. Now, because the castle wasn't prepared for any of this, all of the guards will be ordered to run around and start producing these new weapons right away. So they were given a message and reactively responded. But no one actually came and threatened the castle, so eventually the guards surrendered their posts and continued on with their normal day-to-day tasks as they assumed this was just a false alarm. But then, the same good knight returns sometime later and relays the exact same message. Now the guards are very sure that it is going to happen this time, so they work extra hard to prepare for this incoming chemical attack and make twice as many new weapons, running around and causing chaos within the castle walls. Once again, however, there was no threat of any chemical attack, so the guards put away and stored their chemical weapons, just in case the threat ever does truly come. Once again, feel free to pause the video here to match up this analogy to what might be happening with the COVID-19 mRNA vaccine. From this analogy, you can see that the army of guards prepared special weapons even though they never physically encountered the new chemical, but instead they were able to prepare for the attack by reading a set of instructions from the good knight. The chaos that was created by the army was a reaction to receiving the message, just as how the symptoms you may develop after vaccination are a result of the immune system reacting to receiving the foreign set of instructions from the mRNA.
I got the COVID-19 vaccine, but I somehow still made my little sister sick. Obviously, it doesn't stop you from getting COVID-19. Hashtag doesn't work.
I get how this could be confusing, but this idea likely stems again from the fact that many people think that COVID-19 and coronavirus are interchangeable terms. So to clear that up, you can still catch the coronavirus, but the vaccine helps prevent you from developing the COVID-19 symptoms. So to remind you, the special weapon, the antibodies you built from the vaccine, binds to the epitopes on the coronavirus to protect the virus from accessing the cells. The entirely coded virus now loses the ability to bind anywhere else, and therefore can no longer infect you. This coding process is called antibody neutralization. However, antibody neutralization does take time. So if you catch coronavirus and aerosolize it before your antibodies can fully code it, you can pass it on to those around you.
The COVID-19 vaccine was created way too quickly. There's no way that it's safe. Hashtag no thank you.
The quick turnaround time for the creation of the COVID-19 vaccine was actually quite revolutionary for scientists. After the SARS outbreak in 2003, scientists had a very good understanding of coronaviruses causing respiratory syndromes. Then, in 2020, when the World Health Organization declared the COVID-19 pandemic, scientists were given more than twice the amount of resources, personnel, and funding to develop a vaccine, which, coupled with their previous SARS knowledge, enabled a very rapid vaccine production. The COVID-19 mRNA vaccine was also developed while following all proper protocols. As a matter of fact, the COVID-19 vaccine trials had more participants and a more accurate representation of the population through age, gender, and ethnicity than most other established vaccine trials, such as hepatitis B. The hepatitis B vaccine had 1/8 the number of participants than the COVID-19 vaccine trials. And participants were only males ages 18 to 36. In addition to this, Multiple companies, such as Pfizer and Moderna, ran their own independent trials to ensure the efficacy and safety of all forms of the COVID-19 vaccine.
I don't understand why there are multiple mRNA vaccines. Aren't Pfizer and Moderna the same thing? Hashtag someone explain.
While both companies' vaccines are made with mRNA liposome technologies, they do have a few key differences between them. There are minor differences in lengths as well as the composition of the lipid nanoparticles surrounding the mRNA, and this accounts for different thermal stabilities and dosing of the vaccine. Pfizer vaccines can be stored at 2 to 8 degrees Celsius for up to 1 month, whereas Moderna can be stored at the same temperature for up to 4 months. In addition, Pfizer requires 28 days between doses, whereas Moderna only requires 21. Finally, Pfizer administers a lower amount of mRNA at 30 micrograms, whereas Moderna administers 100 micrograms of mRNA in their vaccine. Of note, both companies' vaccines have mRNA with specific identical nucleotide changes, which will make a modified version of the spike protein, called 2P, that gives our immune system time to build antibodies against it.
I refuse to get one Pfizer dose and one Moderna dose because I will be less protected. Hashtag vaccines.
Even though the vaccines are different, it's actually safe to mix doses of Pfizer and Moderna because of their similar compositions, side effects, and efficacies. Getting 2 doses of Pfizer, 2 doses of Moderna, or 1 of each are equally efficient at protecting you from getting COVID-19. Even though those tweets brought up some really good points, I think it's time to put away the phone again so that we can go over the rest of the timeline. By May of 2021, the Delta variant appeared and rapidly became the next dominant global threat.
That was scary.
The word variant refers to a mutated virus wherein part of the RNA's code has changed. Remember the ACGUs we talked about earlier? If a C is changed to an A, this can change the virus's entire function. The emergence of new variants is the result of these mutations happening to protect the virus from being detected in the body and enhance its ability to infect. Currently, researchers have identified the following SARS-CoV-2 variants of concern: Alpha, Beta, Gamma, and Delta. All of these variants have mutations in the RBD region, which is the part of the epitope the neutralizing antibodies bind to. These mutations can either change the structure of the spike domain to make it bind stronger to the ACE2 receptors and cause more severe symptoms, or the mutation makes the spike protein bind looser to the antibodies so that it can escape attack and become available for ACE2 binding. Now you may wonder, does this mean that the vaccine doesn't work anymore? No, it definitely still works. It just might be a little bit less efficient for the new variants. Here's a table summarizing the efficacy of Moderna versus Pfizer in protecting us from different variants. Feel free to pause if you'd like to look at this a little longer. So as you can see, the vaccine will still be able to protect you against COVID-19. Although it is less effective against the new variants of coronavirus, it still offers protection against the more serious dangers of COVID-19, like death, even though the chances of becoming ill are still possible. So what does the future hold? Of course, we can't predict that, but scientists are trying to make educated guesses about what path the pandemic may take by using a mix of previous pandemic trends and predictive software. However, it's important to note that these predictions are restricted and altered by the use and availability of vaccination amongst the general population.
We are going to need constant boosters forever. When will this end? Hashtag apocalypse.
Oh my gosh, I really thought I put it on mute that time. Well, as we enter 2022, we can understand how it can be hard to see the end of this pandemic, especially after the creation and administration of vaccines promised a return to normal life. But as previously said, the future is uncertain, and the requirement of boosters is partially due to low vaccination rate and the ability of the virus to mutate and avoid the vaccine, such as the Delta variant. So in short, yes, we will need boosters for the COVID-19 vaccine, but this is not new for the vaccination process. In fact, many adults don't even know that they need certain boosters for vaccines that they have already received. For example, the Tdap vaccine for tetanus, pertussis, and diphtheria has a recommended booster every 10 years. Even some vaccines that didn't originally require boosters now do have recommended ones, such as for MMR and hepatitis B. Considering my phone has already been going off non-stop during this webinar, why don't we all take to Twitter and continue busting some myths?
The COVID-19 vaccine is going to change my DNA. Hashtag I'm scared.
Remember, the COVID-19 vaccine contains mRNA that our cells read to produce antibodies against SARS-CoV-2. The key point here is that the mRNA is a separate molecule that is only seen as a piece of information to read in the body. Now remember, we said it stays in the cells, but that doesn't mean it enters the DNA. In fact, mRNA can't even enter the nucleus, which is the department for storing DNA in the cell. So it is literally impossible for the vaccine to alter your DNA.
Why would I need to get the vaccine if I've already had COVID-19? Hashtag pointless.
Even if you were previously diagnosed with COVID-19, it is still important to get the vaccine. The antibodies you produced were very specific to whichever variant you caught, and the vaccine confers more immunity across all variants. In addition, recent studies have shown that those who got COVID-19 but not the vaccine were twice as likely to become reinfected with the virus than those who had had the disease but were later vaccinated. In addition, those unvaccinated are more likely to pass on the virus to other people than those who have been.
FYI, you're going to test positive on a PCR test if you get a COVID-19 vaccine. Hashtag be careful.
It's not surprising that there's confusion about how diagnostic tests differ from immunity tests. But now that you have some basic knowledge about the mRNA vaccines and the coronavirus, we'll try to break it down. Remember, when you receive the mRNA vaccine, this triggers your immune system to begin producing specific antibodies. When you get a test done that analyzes whether or not you have built up immunity, which in the medical field is called a serology test, this test will take your blood sample to determine if these antibodies in the blood are able to attach to the specific epitope and produce an observable signal indicating the antibody has been detected. However, when you receive a COVID-19 diagnostic test, which is typically done via PCR or antigen testing, this test looks to see if the body contains the virus by breaking down the cells and using a DNA amplification method called PCR. For this method, we get to pick the target of amplification, which would be a specific section of the virus. The PCR will then make several copies of that section of DNA, or amplify it, if, and only if, that section of DNA is present in the body. So in the case of the mRNA vaccine, you only receive the mRNA corresponding to the specific target DNA, which means that after vaccination, you will not test positive on a PCR test because that section of DNA is not available naturally for amplification.
I want to have children in a few years, so there's no way I'm getting the vaccine. Hashtag infertility, hashtag vaccine.
I totally understand why you might think this. Near the initial release of the vaccines, myths were circulating that they can cause infertility. However, we can assure you that this is not possible. Scientists were worried that the receptors on the placenta of pregnant women were too similar in shape to the spike protein on SARS-CoV-2. Therefore, there was a chance that the antibodies we create by getting vaccinated could target those receptors. However, this has been disproven, and the Center for Disease Control has said that it is safe for pregnant women to get the COVID-19 vaccine. In fact, they encourage this because COVID-19 can cause them to become more ill than the average person. Whew, that was a lot, but thank you for coming on this journey with us. Today we covered what mRNA is and how it can be turned into a vaccine, the history of mRNA vaccines, coronavirus and the COVID-19 vaccine, differences between Pfizer and Moderna, and the current myths surrounding the pandemic. We really hope that we helped lay out the important information regarding COVID-19 vaccination so that everyone feels completely comfortable reading about the disease and its related scientific terminology, as well as interpreting which statements regarding the vaccine are true or not.